Wow, well, that was quite an introduction, Mike. I really appreciate that. I'm honored, and I only found out that you were going to be introducing me like a couple days ago. So thank you very much for that. No pressure though on saying that I'm funny. So I'm going to have to try to come up with some things here. But as Mike said, my name's Jimmy Bennett, and I'm presenting Grand Rounds today. I just wanted to say it's an honor to be here. I did, in prepping for this, I went back and realized I had given Grand Rounds in this seminar in 2017, and I looked back at those slides and realized that we made some progress in understanding somatic mosaicism and vascular anomalies, but a lot of the questions remain unanswered, which I think means we're just asking good questions. So I will take it away. So the objectives for the talk today these were for the CME, or for you guys here and for the audience at home, to recognize the limitations of blood-based genetic testing for individuals with vascular malformations, most of which are due to mosaic activating mutations, to describe the two main factors that go into successful molecular diagnostics for vascular malformations, which is the use of affected tissues, FFPE or frozen, as well as the use of high-sensitive methods that can detect low varying allele fractions. And then also, just more broadly, I really wanted to try to expand, not just to talk about vascular anomalies, but just more generally, to consider the implicit assumptions that happen in almost all genetic tests for non-neoplastic disease. So I'm going to start just to increase engagement here with a poll question. So this is a kid, this is a patient at Seattle Children's, full-term male infant who was born with this flat red macula patch in the segmental distribution on the left shoulder. And presented in heart failure, actually. We had a nurse practitioner at Seattle Children's who I think saved this kid's life, sending him to the ER. And you can see why he had heart failure because of these direct artery to vein connections, just massive overgrown vessels in the left arm. There's no family history. So the question for you to ponder, and I'm not going to answer it yet because I want everyone to pay attention, okay, is what testing, if any, should be performed? So do you think the best test would be to get a targeted high-depth sequencing panel from the kid's blood, targeted high-depth sequencing panel from a skin biopsy, genome sequencing from the blood, exome sequencing from cultured fibroblasts from his affected skin, or no testing indicated, this is non-genetic? So ponder that for a second. Okay, the outline of the talk today is here. I'm going to start off the first part. I'm just going to, in general, build the stage for the importance of new mutations in clinical genetics, and then I'll get into the VANSeq as well as some future directions for somatic mosaicism. So, I know this word precision medicine is an exciting new word, but I do always like to point out that precision medicine is not new. We've been practicing it for hundreds of years in pediatric genetics. And this is a representation of the history of pediatric genetics. All of these children, all these pictures are of kids who've had some congenital malformation or developmental issue. And it was long recognized based on clinical features, facial patterns, what's typically known in genetics as dysmorphology, that you could gather predictive information about a child's future, about recurrence risk, etc., from looking at their face. I really like to show this in all of my talks because the textbook for clinical pediatric genetics is called Smith's textbook, and there are several others, and it frequently shows children with rare genetic diseases that are, naked in front of a grid. Sometimes they're dead, and I just really want to point everyone to this awesome website called Positive Exposure, which is operated by this gentleman, Rick Giadotti, it's just an incredible resource for learning about pediatric genetics and dysmorphology, and a much more positive slant on real people and kids with these diseases. And so the story is he was a model. He was a photographer for models in New York City, and he got sick of taking pictures of sort of pouty models. And he met this one woman who had albinism, and she was very shy and kind of hidden up, and he took photos of her, and she kind of came out of her shell. And so now he does this. He goes around, and I was actually able to attend one of his sessions with patients with Coffin-Siris syndrome. So it's pretty amazing to be in a room with 30 kids who have a rare disease. And it also helps you as a geneticist in terms of syndrome recognition. But really, genetics, that's the history of genetics. I think that currently rare disease in genetics is really represented by this picture. So, this the trio exome sequencing revolution, starting with, decreasing costs of next generation sequencing circa 2010. And the model, quite simply, is this. We take blood from mom, dad, and kid. We do some sequencing. We sequence all the genes, or you can change slight flavors of that, but that's the bottom line of it. And in many different studies for children with, structural birth defects, sort of syndromic presentations, the diagnostic rate of exome or genome sequencing is approximately 25 to 50%. And this is really driven by new mutations. I really want to make this point here. So these numbers here show via Illumina short-read sequencing the number of coding, so in genes, single nucleotide variants that are inherited per genome, approximately 22,000, versus new ones that are not seen in mom or dad, which is about 1 to 2. And what I think is really interesting to think about is that, when you sort through all these variants, those 1 to 2 drive over 50% of diagnoses of rare pediatric diseases. The 22,000 you have to sort through to find recessive conditions or inherited autosomal dominant or X-linked conditions. So these new mutations really do have a huge impact on clinical diagnostics. And the reason for this is, there are several reasons, but new mutations can happen all the time. They certainly happen in the sperm or the egg prior to fertilization, but they can happen after fertilization. And that's really the main lesson I think I'm trying to get across here today. If they occur during early human development, and it's represented here by this pink lightning bolt, you end up with an individual who's a mosaic, a mix of different genomes that are derived from the same zygote. And I'm going to put the picture of this woman who's the lab manager in my lab, Dana Jensen, up probably several times. She's had an enormous impact. There was actually a BBI story about her. She's an amazing individual, and she gets credit for this drawing, which is now just like, so many people have used this drawing now. We probably should have copyrighted it. So key concept number 1 is that sequencing one tissue from an individual, just because it's convenient, blood or saliva, is not going to identify these types of mutations. And really, this concept of mosaicism challenges this idea that blood-derived DNA is identical to all their cells. I mean, this is an implicit assumption when you have a blood sample and someone gets their personal genome. And just to do a little bit of math here on this, you start off your existence as a single cell. By the time you're born, you're 10 to the 13th or 14th cells. And probably at least one mutation occurs with every single cell division. So really, every base in your 3.2 billion genomes is probably mutated in at least one cell in your body, and every multicellular organism has more than one genome. So if we go back to the question I posed sort of at the beginning, why is the diagnostic rate of genome sequencing only 25 to 50%? Why not 100%? There's a couple of reasons for this, and this is an active area of human genetics. Certainly non-coding variation is something that is understudied, and there's a lot of variants that are not in genes that could contribute to disease, and people are working on that. You can have variants that are in 2 or more genes, complex genetics. That's important and very hard to sort out. There can be variants that are present in every cell in their body, but they're not currently detected by methods. Short-read sequencing doesn't detect certain types of variations. We often forget about the fact that congenital malformations can be caused by viruses and environmental effects and things. Not every birth defect is genetic. But a simple one, I think, the simplest maybe in all this list, is simple that the variant is not in blood. And that goes back to this idea of mosaicism. So, and I recognize that I'm speaking here to a Department of Lab Medicine Pathology with a heavy focus on cancer. And I think, everyone recognizes this phenomenon in cancer, that you have a genetic change in a cell that proliferates, more changes occur, et cetera, et cetera. You have this, progression from adenoma in crypts to ultimate, carcinoma. But it remains still less well recognized outside of cancer. And I think, like in pediatric genetics, when I go to pediatric genetics meetings, there's this feeling of an arms race that has happened in diagnostic genetics. You start off with targeted panels. If that's negative, maybe we can sequence all the genes through an exome. People say if the exome is non-diagnostic, maybe we should sequence all 3.2 billion bases of the genome. And now, there's a lot of interest in even more exotic, diagnostic methods, looking at long-read sequencing, looking at epigenomes, or looking at RNA sequencing. And these are all terrific, things. All these need to be done. But and then I also wanted to make the point that there's this term that's in society about personalized medicine, your personal genome. Companies are always talking about it's in our DNA, like as if one individual has one personal genome, that's it. To a first approximation, I think that's true, but it's not always true. And I think that, I just would add to this arms race the possibility that mutation is just not in the blood. So with that introduction past me here, I'm going to move on to talking about vascular anomalies and VANSeq in more particular as a poster child for mosaicism. So I like to put this picture up. These are all different children pictures of children and 2 pathology slides. Again, this is a pathology conference. I had to put in some slides of tissue. And you can see these are all vascular lesions. If you look at them under the microscope, you see too many blood vessels. They look bloody. Clinicians typically would call all of these things hemangiomas, which is not very precise or useful. And so I often like to ask people, like, you see this excess proliferation. Sometimes you see excess extra mitoses. These things can sometimes spread and grow. Is this a tumor or a congenital malformation? And this is kind of, it's not, there's no answer to the question. I mean, it's supposed to be sort of challenging. Vascular anomalies is a term that refers to any sort of abnormal development or presence of blood vessels. They can be separated generally into 2 large categories. So when someone says a vascular anomaly, they are referring to one half of these are known as vascular tumors, which typically have the suffix oma. So infantile hemangioma, caposiform hemangioendothelioma. Those would be vascular tumors. In general, this is an oversimplification, but the way you can keep it straight in your head is these things are very proliferative. They tend to grow even postnatally. They tend to not be present in the first trimester of human development. They're not developmental abnormalities. They're proliferative. And if you look at them under the microscope, you can see many mitoses. As opposed to vascular malformations, which are really which is really aberrant morphogenesis of either blood or lymphatic vessels. When you look at them under the microscope, you do not see many mitoses. And these are present in the first 5 weeks of human development during early vasculogenesis. So this is an example of an infantile hemangioma on the left and a capillary malformation on the right. More specifically, the International Society for the Study of Vascular Anomalies has broken these down, and I'm not going to go through the complete classification of vascular anomalies here. But in general, the vascular tumors, the major ones are listed over there on the left, and they can be organized from more benign ones to more aggressive ones. And then the vascular malformations are generally split into high flow lesions, which means you put an ultrasound on the thing and you see a lot of high flow moving through it, or low flow lesions, where an ultrasound does not show a large amount of flow. The high flow lesions refer to arteriovenous malformations, which are these direct communications between high pressure arteries and veins. And the low flow lesions would include abnormal proliferation of lymphatic vessels, of what appear to be more venous vessels or capillary vessels. So this is the sort of rough classification of vascular anomalies. I'm not going to go into a huge amount of detail on this because I want to get to the diagnostic part of it. And in fact, for the rest of the talk, I'm really just going to focus on 3, the most common and the most clinically important isolated vascular malformations. And these are lymphatic malformations, venous malformations, and arteriovenous malformations. So on the far left, you see 2 pictures of children with lymphatic malformations. On the top are pictures, and the bottom are their corresponding MRIs. You can see this baby, on the far left with the sort of growth, above her shoulder, and it's a little bit harder to see, but that child that's next to her has a bilateral lymphatic malformation sort of encasing the airway and has a tracheostomy in place. So lymphatic malformations are one class, venous malformations are another. Here you can see this child just has a little bit of a bump on his neck, and you can see the corresponding image here in the MRI. And this child has this bluish vascular venous lesion in his hemitongue, and you can again see that in his MRI here. And then lastly, here's an example of an arteriovenous malformation, which is this warm pulsatile lesion. If you put your stethoscope on it, you can hear a pulse or a bruit. And then on angiography, you can see this early blush of dye in the venous side without having traveled the amount of time through capillaries. So in general, all of these things can cause medical problems by compressing adjacent structures. They can affect swallowing, feeding, breathing. They can cause pain and disfigurement. They can cause bleeding and coagulopathy, infections. Particularly AVMs, which are really clinically the most severe of the 3 types of vascular malformations, can lead to local tissue ischemia, even heart failure because of overcirculation. And traditionally, the treatments have been surgical resection or sclerotherapy, which is where you inject some sort of a toxin into the lymphatic cyst or into the vein to cause the endothelial cells to die and sort of shrivel up. They don't generally run in families, and this is sort of the focus of VANSeq and the rest of my talk here. So a key concept, I've sort of gone through and listed the key concepts. This is an example of a child with a large facial lymphatic malformation, left cheek and neck. And if you were to sequence blood from this individual, you would never detect the mutation, no matter what fancy genetic testing you do. You will detect the mutation if you look in the tissue. So that's a key concept that I hope is a take-home, that in most individuals with isolated vascular malformations, you're never going to be able to detect the mutation on routine analysis of blood cells. And just to summarize, quite a bit of work, but in a single slide, but it's not too hard to do. We now know that, and this is a summary about past 5-10 years of work, that mosaic activating mutations in 5 oncogenes, which will be familiar to people who work with cancer here, are the cause of these 3 main classes of vascular malformations. So activating mutations in the phospholinositol kinase PIK3CA, as well as less frequently V600E mutations in BRAF are causes of lymphatic malformations. Mutations in the receptor tyrosine kinase TEC, as well as PIK3CA, can cause venous malformations. And then activating mutations in BRAF, KRAS, or the MAP kinase, MAP2K1, can cause arteriovenous malformations. And what's amazing about these discoveries is that all of these genes are in the same pathway. This is a well-known pathway that stimulates growth and proliferation of cells, among other things. All of these genes are in the same PIK3CA, RAS, MAP kinase pathway, which is drawn here, an oversimplification, of course. And here just sort of shows the corresponding genes that have been implicated in causing venous and lymphatic malformations, as well as the genes that have been implicated in causing arteriovenous malformations. So what? A lot of pediatric genetics is about recurrence. There's a whole arm of pediatric genetics and in lab medicine. If it's not syndromic, if it's not running in the family, what good does a genetic diagnosis do? Kid's got an AVM, kid's got an AVM. If we know it's due to KRAS, who cares? Obviously it's a straw man question I'm asking there, because the thing is that since this pathway has been heavily studied and heavily, by people who are interested in treating breast cancer and colorectal cancer, which, many of these genes, the exact same mutations, can cause these cancers, the drug companies have long targeted this pathway. And so, only in the past few years has it been recognized that we can actually repurpose drugs designed for cancer to treat developmental malformations. That's kind of an amazing thing. There was I want to really make sure that point is emphasized, because developmental malformations, there was a thought at one time that these happen in the first 5-6 weeks of development. The only thing we can do by the time the kid's born is surgery. There's not ongoing abnormal signaling, but in fact there is. Of course, Novartis and the drug companies do not care very much about vascular malformations, but they certainly care about breast and colorectal and other common cancers. And so here's an example of a child who's been on this PIK3CA inhibitor known as alpelisib, which was FDA approved for PIK3CA positive breast cancer under the name, I forget the name of what it's called for breast cancer, but for the pediatric indication it's called Vijoice. And you can see this dramatic improvement in the lymphatic. This child has a little bit more than an isolated lymphatic malformation, but this child does have a PIK3CA related overgrowth that includes vascular malformations, and you can see this improvement on this drug alpelisib. So that's key concept number 3, that we are starting, one of the justifications for identifying these mutations, these somatic mutations in non-neoplastic disease, is that it can help us, make kids eligible for MEK inhibitors, for PIK3CA inhibitors, BRAF inhibitors, and this is a lot of mTOR inhibitors, also like sirolimus, and so some of those drugs are shown here on this pathway. Okay. So one of the challenges, however, in diagnostics for somatic mosaicism is detecting low-level variants. Okay, this is a major challenge. So I already mentioned one of the challenges, we got to get the tissue. Okay, the other challenge is we have to have a method that will detect these low-level variants. So this is a scheme here to introduce the concept of variant allele frequency, or VAF. So on the top is an example of a heterozygous mutation. The mutation is in yellow. We have 10 cells up there, and the cells are diploid. They have a yellow copy of a gene and a blue copy of a gene. So I think people are pretty used to, if you ever looked at next generation sequencing and you see an inherited variant, it is present at 50% levels, because half of the DNA molecules have the mutation and half do not, okay? However, when you have somatic mutations, only a small fraction of the cells will have the mutation. In this case, 2 out of 10 cells have the mutation, which would be 20% of the cells, but the variant allele fraction 2 out of 10 would be 5% of the cells, but the variant allele fraction is twice that, or 10%. because each of the cells has 2 copies of the D. So I want to make sure people understand this concept. And so what it turns out, and this is the research in my lab and others, when we look at individuals with lymphatic malformations, but the same is true for venous and arteriovenous malformations, we see that the typical detected pathogenic PIK3CA mutation that we're detecting is typically present at 0.1 to 13% variant allele fraction. The implication of that is if you double those numbers, 0.1 to 13%, it means that 0.2 to 20% of the cells in the lesion have the mutation. Most of the cells in the lesion do not have the mutation. That's key concept number 3. Despite these large, grapefruit size or bigger lesions, most of the cells in these tumor-like non-neoplastic masses do not possess the mutation. So this is not like cancer. I mean, cancer, you get a tumor out of a patient. It's not, I'm not a pathologist, so it'd be very difficult for me, but you can assess the tumor purity of a given slide. You can look and say, oh, there's a lot of blue cells in there. That's the limit of my understanding of it. But you can't do that for these. When you look at all these tissues under the microscope, it's too many and disorganized, but they express all the same markers of lymphatic tissue or blood vessels. You cannot detect. There's not an abnormal cell type that's easily seen there under the microscope. So I think that's really important, that you have to use a method that can detect variant allele fraction down to 0.3 or 1%. And some people have used cancer assays like Oncoplex or similar type assays for vascular malformations. The problem with that, one, is gene content. But another problem is that the limited detection for many cancer assays assume that you have a cancer purity of at least 5-10% in going into the assay. And so really I think the vascular malformations need sort of specialized testing. And so for this reason we developed this test VANSeq that Mike mentioned already. It's not rocket science. All we did was we took a good list of genes that are recurrent, about 47 genes, and we validated it to accept many different tissues, not just blood and saliva, which is the main thing in pediatrics, but also FFPE and tissue. So those are the 2 key ingredients: high-depth sequencing, as well as the ability to sequence affected tissues. And just to summarize, it's been a pretty successful test. So we launched it in 2019, and you can see the number of signed out tests here year by year. As of Halloween this year, we've done a total of 848 individuals, and the volumes keep increasing each year. So it's been successful, and I think it's had a big impact on molecular diagnosis of vascular anomalies in the United States and beyond. Here shows where the tests have come from. So you can see we've had tests from Alaska, Hawaii, Uruguay, and as far away as Pakistan, and about 3/4 of these tests now come from outside institutions, and 1/4 from internal. So I mentioned the 2 secret ingredients of this successful test. One was high-depth sequencing and ability tissue, but really the most important ingredient is here, which is this amazing team that we have. I mean, it really does take a village, and so I didn't want to wait to the end to thank all the people who did this. And so our directors, Naya Nelson, Kate Paschal, Annika Wandler, as well as variant scientists Maddie Gillentine, Sophia Bhatia, and Candice Myers, who really, I have to call her out in particular because she had a major role in helping to develop this test. And it's not just the test. I like to say the patient sequencing DNA is the easiest part. Staffing the front desk, having triaging samples and things like that. So Darcy Stern and Susan Showalter are lab genetic counselors who do an amazing job. And then, of course, we're not just building this out of scratch. We already had a pre-existing incredible vascular anomalies team, clinical, led by a head and neck surgeon, Jonathan Perkins. So, I mean, really, it wouldn't be, and now a genetic counselor who sees patients who's stationed in vascular anomalies, Zoe Nelson, is just in addition to every other person in the Seattle Children's Molecular Lab. So, just to show you some of the results of VANSeq. We have a pretty good diagnostic rate. 60% of the variants, 60% of the cases have a pathogenic or likely pathogenic variants, but with a very low VUS rate, only 2%. And here you can see about 45% of the positive cases have a variant allele fraction of less than 5%. So this is a sort of a showing of all the pathogenic or likely pathogenic variants that have been reported. You can see these are the ones that have variant allele fractions between 0 and 5%, 5 to 10, etc. And you can see the vast majority, or almost 50%, have a variant allele fraction of less than 5%. And these are diagnoses that are helping these kids become eligible for medications. This is a figure that Candice Myers put together for a previous talk. I just wanted to show one of the strengths and one of the advantages we have, as opposed to maybe some other disorders, that so many of these mutations that drive these lesions are recurrent. They're somatic activating mutations, so they're not loss of function mutations. You know, with loss of function mutations, the mutation can be anywhere in the gene. Here, these are a little bit more, you know, they're recurrent. And so you can see this is a graph of showing a number of pathogenic and likely pathogenic variants. And you can see that 70% of them are recurrent in pink. So most of the variants that we're detecting are variants that have been seen before. So variant interpretation is not hard for VANSeq. These are known variants, PIK3CA, H1047, BRAF, V600E, famous mutations. But the challenge more is it real when we detect it or not? And then here is this is a slide just showing the difference in the diagnostic rate from when we get tissue, where we're at about 68%, for when we get another specimen, where our pathogenic variant rate is only 35%. So it really is important to get the tissue. And I can't stress this enough. I mean, we go to meetings, we do this education, we still get orders from patients with isolated vascular malformations, and they send us a blood sample. I mean, you might as well throw it in the trash. You know, I mean, it's not very useful. It's the tissue's the issue. And I want to say I want to give a specific shout out to Dr. Nia Nelson, because she's a molecular pathologist who joined the team and has just been an incredible addition to the team. Not only is she able to sign out molecular tests, but she's a true pathologist. And you know, when VANSeq first came out, you know, I'm not a pathologist, and so I would get these questions about, well, which blocks should we use? What areas? You know, and I couldn't answer these questions. This is from a presentation she gave recently at our Vascular Anomalies retreat of a child with I'm not going to go into the details, but the child had a possible PIK3CA mutation, was very sick, was having a lot of necrosis and infection in their skin, and the surgeons kept going in and doing these debridements and getting biopsies, and every time it came out, it was kind of we couldn't get any DNA out of it. And so, you know, Nia is able to look at this slide and say, oh, there's the vascular malformation. Everything else is necrosis, infection, and inflammation, you know, so you know, it's just an incredible person to work with, and she's just brought a lot of value to VANSeq. Okay, so I've told you that the tissue is the issue here, but what if the tissue is not available? You know, and this is, comes up with cancer again. I mean, I think the one theme of this talk is that all we're doing is applying everything people have done for cancer and just applying it to non-neoplastic disease. So, you know, kids can have a vascular anomaly, it can be a dangerous location. Here's a picture of a vascular malformation encasing this child's airway. Maybe that's not a great place or a hard place to do surgery. You can have important structures like the facial nerve, which are in the face. The kid can be too young for surgery or too sick for surgery. And then sometimes it's not always clear what tissue to test. There are I'm showing you a lot of examples where it looks pretty obvious what the affected tissue is, but that's not always the case. So what if the tissue is not available? And so what we did was, again, taking a page from oncology, we looked at cell-free DNA. So I'm pretty sure this crowd is familiar with cell-free DNA, but just to review, if you take blood sample and you let it sit for a little while or you spin it, you get three different layers. You get the red cells at the bottom, which do not contain any DNA. You get the buffy coat in the middle, which is the usual DNA. That's the white cell-derived DNA. And then on top, you get this watery stuff that's referred to as plasma. It turns out there is free-floating DNA inside that plasma. And it turns out that about 10% of cell-free DNA in healthy individuals is derived from endothelial cells. And so we thought, well, since these diseases are vascular and endothelial cells play a role, can we make any of these diagnoses in kids who haven't had surgery? And so here's the methods of this study that I'm going to share with you. And this was done by the members of my research lab. We collected 36 individuals with venous malformations and 28 individuals with arteriovenous malformations. We collected, in addition to collecting tissue from them, all of them had known tissue-based mutations. We also collected blood in a cell stabilization or STREC tube, which is a good tube for cell-free DNA. We collected either peripheral blood, just from a peripheral IV, or in some cases, if they're going for interventional radiology, they're getting sometimes the interventional radiologist will percutaneously stick a needle into the vascular malformation itself. And so some of those were lesional blood samples as well. And so in this case, we knew what mutation we were looking for. We had the tissue-based mutation. And so then we just used a sensitive method called ddPCR, or we did high coverage sequencing to see if we could detect that mutation. So our results are here. We sort of defined the tissue-based mutation on the bottom. That's the denominator, would be your gold standard. And we just said, could we detect this in the plasma? And so for venous malformations, we detected the known either TEC or PIK3CA mutation in about 47% of the samples, 17 out of 36. And for arteriovenous malformations, it's about 25%. So still, obviously, tissue is the gold standard diagnostic analyte, but this is suggesting that if your patient has not had a surgery or you can't get tissue, that it is not unreasonable to go ahead and give these a try for venous malformations and arteriovenous malformations. Now, I mentioned that we detected blood samples both peripherally and centrally. And so, sorry, this is kind of complicated plot, but I'll show you. Every single line here represents an individual patient in the study. And at the top shows whether we were able to collect lesional blood from them. We separated the lesional blood into plasma, or we also examined the blood cell pellet from the lesion. And then we did the same thing for peripheral blood. You can see it's a lot easier to get peripheral blood. And the bottom shows sort of the patient ID, and then here are the different mutations. I'm not going to go into detail about the mutations. Let's just say they're either mutations in the receptor tyrosine kinase TEC, which causes venous malformations, or they're mutations in PIK3CA. But the bottom line of it is that our sensitivity for detecting these mutations in cell-free DNA was higher in lesional samples. And I'm highlighting these 5 samples here where we had both a good peripheral blood sample from the patient in which we were not able to detect the mutation. And then you can see up here is showing in these samples we were able to detect it in the lesional sample. So I think there's a real sort of clinical lesson here, which is if you have a patient with a venous or an arteriovenous malformation and they're going to interventional radiology procedure, you should collect a Streck tube of blood. And so, it's kind of interesting because interventional radiologists are not the traditional providers who are ordering genetic testing, and that is something we're having to work on. But yeah, this is the sort of the conclusion of the study. Now, going back to this patient I mentioned at the beginning with the arteriovenous malformation. So this patient has an activating mosaic mutation in MAP2K1. We're able to detect that from the skin biopsy. That was the answer of the question. And so we thought, well, without going into a great deal about this patient's clinical history, because of this MAP2K1 mutation, they were placed on a MEK inhibitor. And this was done primarily because this child was having a lot of necrosis and ischemia at the tips of his fingers. And so what you can see here in this graph along the x-axis is the time elapsed since he was started on this MEK inhibitor, trametinib. Here's time 0 here when he was started on trametinib. And we were able to get 5 different samples from this patient peripherally. And in each sample, we were able to measure the variant allele fraction of the known MAP2K1 mutation. So you can see prior to starting the drug, it was about 3%, 2%, 1%, about 2%. So in this N of 1 sample, it was not a useful biomarker because the corresponding clinical pictures, he did respond to the MEK inhibitor. The ischemia and necrosis of his fingernails definitely improved on the MEK inhibitor. So, I mean, we are very interested in, as are many people, if cell-free DNA can be used as a biomarker. I do think there's a difference here in how we're using these drugs compared to cancer. In treatment of cancer, the goal is to eradicate the mutant cells completely. And the doses are really to kill those cells. The doses that are used for treatment of vascular anomalies are a little bit lower. And I don't think that the goal is we cannot eliminate all of the mutant cells within this child's arm. And so it's a little bit of a different situation. So, I don't think we can make a conclusion here other than to say it is feasible to try to do things like this, and more study is needed. Okay, and then lastly, I want to mention how lymphatic malformations are a little bit different. So unlike venous and arteriovenous malformations, which are connected to the peripheral circulation, lymphatic malformations frequently have this cystic structure, and they are not connected to the peripheral circulation. But these cysts are full of cystic fluid. Here you can see this giant balloon-shaped lymphatic malformation in this child, and this is just liters of this stuff. This straw-colored fluid can be drained. And so with that observation, as well as with the fact that the vast majority of children with lymphatic malformations have PIK3CA mutations, and they have them in one of 3 locations. They're recurrent. These are these hotspot mutations that are the exact same hotspots seen in breast and colorectal cancer. These are at codons 1047, H1047R, E545K, and E542K. There are some non-hotspot mutations, but these account for the vast majority. And so, again, here's Dana Jensen in the lab. She developed a multiplex ddPCR assay. And so this is I'm not going to go into a lot of detail about how the assay works, but it has a very low DNA input requirement, and it can, based on the location of these clusters, detect the presence of 3 different alleles in PIK3CA, of the 3 different hotspots. And what she was able to do was take cell-free DNA from cyst fluid and basically show that the diagnostic rate and the distribution of mutations was about the same as it is from tissue. So unlike for venous malformations and arteriovenous malformations, where tissue really is superior to plasma cell-free DNA, In this study, cell-free DNA from cyst fluid was comparable to the lymphatic malformation itself. So because of this, we have just recently launched a new assay at Seattle. I mean, this is kind of big because, again, same thing. You know, kids with lymphatic malformations, they want to be. There's a lot of demand now. Can I put my kid on these new drugs like alpelisib? Okay, but in order to do that, we need to document the presence of a PIK3CA mutation. This is sort of companion diagnostic kind of idea. And so not all of them are able to go to surgery, or they want to potentially start the drug prior to surgery. And so on the basis of this research, you know, we launched this test. We call it the SMaHT test. This is the first cell-free DNA-based assay available at Seattle Children's. And really the primary indication is multiplex PIK3CA testing for lymphatic malformation cyst fluid. I mean, I wouldn't call this non-invasive. Obviously, you can go to any phlebotomy and get a peripheral blood draw. This requires a surgeon or someone to, with ultrasound guidance, stick a needle into a cyst. But it's certainly a lot less invasive than taking a kid to the OR. So far, I will say, we're at 3 out of 9 diagnostic rate, which is a little bit lower than what we did in the research lab. And so I'm not sure what the reason for this is yet. I think this is an interesting thing. You kind of get some results on a cohort research, and then you bring it into clinical lab, and things change a little bit. I have noticed that the volumes of fluid that we seem to be getting clinically are a little bit lower than the ones that we were getting research, and that may be playing a role. But anyway, that's where that's at. And again, I have to, I don't think I remember to put her picture here, but again, I have to mention Candace Myers, who's here in the audience today, because she was the architect of this assay and performed the validation, and is really just, we wouldn't have this assay without her. Okay, so to summarize this part here, I've told you that routine blood-based DNA sequencing misses disease-causing somatic mutations and vascular malformations. I've told you that common isolated vascular malformations are caused by recurrent activating mutations in oncogenes of the PI3K RAS-MAP kinase pathway. I've told you that clinical diagnostic testings for these isolated malformations is providing justification for targeted pharmacologic therapies. And I think most importantly, I've told you that most of the cells in these large lesions do not possess the mutation. And lastly, the success of the VANSeq assay is due to its high depth of coverage, the use of affected tissues, and just an incredible team of people to work with. Okay, I also, to summarize the cell-free DNA part, I mentioned that cell-free DNA is a useful diagnostic analyte for vascular malformations, that cyst fluid from lymphatic malformations can identify the PIK3CA mutation in the majority of them. A couple of things that I didn't have time to mention, but I'm just saying here, that the cyst fluid VAF, variant allele fraction, is comparable to what we see in tissues. When we look in peripheral blood for lymphatic malformations, we never see the mutation, and I think this makes sense because lymphatic malformations are not connected to the peripheral circulation. And then finally, we launched this SMaHT assay on the basis of that work. And lastly, I told you that plasma-based cell-free DNA can identify the known tissue-based mutations in about 50% of venous malformations and 25% of arteriovenous malformations. And I mentioned a story about cell-free DNA having potential as a biomarker for assessing treatment in vascular malformations. Okay, so I'm going to leave vascular anomalies here a little bit, or the clinical vascular anomalies for this final part of my talk. And I hope everyone now recalls that the right answer here would be targeted high-depth panel from an infected skin biopsy. Okay, so I wanted to tell a little bit about some of the big questions about vascular malformation, somatic mosaicism that are unanswered. Some of these things are things I've been thinking about for a while, and we still don't have answers. So the million-dollar question for vascular malformations is, how do you get such a large lesion when the 95% of the cells in this child's lymphatic malformation are wild type? And so we don't exactly know where the mutant cells are in this lesion. We know endothelial cells are primary, but we don't know exactly where they are. And as I said, how do such a small number of cells result in such a large lesion? There must be intralesional or paracrine signaling, this is unknown. So again, I just want to make sure I mention and thanked all the people. This is the people who've worked in the research lab, and I've got pictures of a few of them in the slides to come. But I just wanted to say they are also a great group of people to work with, and we kind of have this nice thing where we kind of work back and forth with the clinical lab and the research lab, and it's been, I think, just we've made a lot we've had a lot of progress, and it's been a lot of fun. I get to work with a really great group of people in the research lab as well. Okay, so as far as what we've done to try to address this question of where the mutant cells are, we know from other studies that laser capture microdissection of endothelial cells in vascular malformation tissues when you capture the endothelial cells, you can enrich the mutation. So clearly the endothelial cells are concentrating the mutation. It's also known that if you take these vascular malformations and you do things like in vitro culture of the endothelial cells, you can enrich for these mosaic mutations. It's also been shown that using mouse models where you drive these mutations using endothelial cell specific drivers, you can recapitulate some of these mutations. So the endothelial cells are clearly important. But does it look like this? So so here is a picture of a lymphatic malformation. These are the endothelial cells. This white space is the cyst. Is it the case that all of the lymphatic endothelial cells have the mutation and none of the stroma, the fibroblasts, the smooth muscle cells, and none of them have the mutation? Is it the case that maybe the mutation is cyst by cyst, like this cyst has the mutation, but this cyst over here does not have the mutation? That's a possibility. Maybe it's sort of salt and pepper, that there's 5% of all endothelial endothelial cells in every cyst has the mutation. Or it's possible that maybe it's in endothelial cells, but it's also in non-endothelial cells in some of the stroma. So this is not known. So one of the things that we've been trying to do, and I don't have results for you to do this to show you this yet, but we have been trying this assay called BaseScope, which is an RNA-specific, allele-specific in situ hybridization method. This is some published data from 2 breast cancer cell lines. On the left is using reagents that cause a red dot to occur when there is PIK3CA wild-type RNA, and you can see presence of that in both of these cell lines. And on the right is a in situ hybridization that is specific for the PIK3CA H1047R mutation. And here you can see these red dots are present in the breast cancer cell line that has the H1047 mutation, but not in the breast cancer cell line that has another PIK3CA mutation. So we are in the process of trying this method, or when I say we, I want to mention Dr. Kelsey Loy, Miranda Pham, and Natalie O, who are working on this in the lab. So that's an area of active exploration. Another thing we have been doing is using single cell methods. So this is just a 10x based single cell ATAC-seq plot where we take individual cells and look at the accessibility of their chromatin to try to identify the cell type. And what you can see here is we have isolated about 5,000 cells from a single individual with the PIK3CA E545K lymphatic malformation, and we are able to identify the different cell types. So they're circled here: fibroblasts, fat cells, B cells, T cells, as well as lymphatic endothelial cells, which we can define by their increased expressibility of this master regulator of lymphatic endothelial cells, which is known as PROX1. But what's really cool about this method, and this is in collaboration with Dan Landau's group at New York, is that we're integrating genotypes with this as well. So we know that these cells, we know that some fraction of these cells will have a PIK3CA mutation, and you can see that in blue are the cells that have an identified PIK3CA mutation, and in green are the cells, to be precise, that have a PIK3CA wild type. It doesn't mean they don't also have a PIK3CA mutation as well. And so we're looking at that. And so far it's still early. We don't have a conclusion yet, but we are able to identify the expected cell types. We do see some enrichment of PIK3CA mutations in the lymphatic endothelial cell cluster. The fraction of blue dots here is a bit higher than it is in some of the other ones. But interestingly, we do see mutant cells that are not restricted to the lymphatic endothelial cell cluster. So I think we'll have to see how that plays out. But I mean, one thing that we can use this data to do is if we can increase our efficiency of identifying the PIK3CA mutant cells, we can then look at the genes that they're expressing, look at the secreted ones, and then those would be candidates for intralesional signaling within these molecules. So this is the work, again, of Dana Jensen and Natalie O in the lab have been working on this. Another method that we have used to apply to vascular malformations, which I think some of you may be familiar with, is the nanostring method. I won't go into great detail about this, but suffice it to say, you can use a laser. It's sort of like bulk laser capture microdissection. You can capture different regions and look in general at their transcriptomic profiles. And so here we have done this. We have 3 regions that have been captured here. One region, region 1, which is the lymphatic endothelial cells. You can see we're right up against the cyst here. These are the endothelial cells. A region that's close by to the presumed mutant lymphatic endothelial cells, and a region that's further away. If we can try to think about, can we see differences in signaling or gene expression based on their proximity to the lymphatic endothelial cells. It's still early days. All I can say here is that if you look here in this picture, we do see some enrichment. If you look at this column versus this column, you can see that the lymphatic endothelial cells do show an enrichment in PROX1 expression, but more analysis is in progress to really make some conclusions from this line of data. And this is work that's being done by Kelsey Loy, Miranda Pham, and Nia Nelson. Okay, and so in the very last bit here is, what's so special about vascular malformations? I've told you all this stuff. So everyone in this room, I think, is familiar that post-zygotic mutations cause cancer. I hope that you're now aware that post-zygotic mutations cause vascular malformations as well. But what about congenital heart defects? What about autism? What about just, all the other disorders that are not often diagnosed by routine blood-based exome sequencing? I don't know that there's really anything special about vascular malformations. I think it's lower-hanging fruit. I think recurrent activating mutations are easier to identify. I think these are a group of patients that routinely have surgery, and we can get at their tissue, so that's very helpful. But this is just a summary of data from Tim Corrins, who's at the Broad Institute, that has demonstrated that across many different normal tissues, there's a huge amount of post-zygotic mutation. And in fact, somatic mutations accumulate 10 to 100 single nucleotide variants per cell per year. You can see that the rate of accumulation of single nucleotide variants is highest in the placenta and lowest in germ cells. And so, I don't see that there's any reason that at least some of these variants couldn't be contributing to other diseases that are not diagnosed by routine blood-based sequencing. And so one question is, could hidden somatic mutation be contributing to other structural birth defects? And again, this is still early days for this project, but we have decided to look at this one disorder called craniofacial microsomia, which is a fairly common structural birth defect in children with some facial asymmetry. You have typically underdevelopment of the mandible, maxilla, the ear, and the orbit. It does not typically run in families. And although there are some exceptions, there are cases where blood-based diagnostic testing can identify mutations. There are rare cases where it runs in the families. That's not usually the case. And it's also a disorder for which we have a very strong clinical group at Seattle Children's, and it's a disorder for which tissue is available. And so what we've done is we've taken a trio, an individual with craniofacial microsomia, and we've done very high coverage exome sequencing on tissue from this individual, preauricular tag, which is a fleshy bit of tissue in front of the ear that's frequently seen in these individuals, as well as high depth exome sequencing on this patient's blood, as well as both parents. And then we're generating candidates for looking for non-inherited variants that are specific to the patient's tissue and specific to the patient's blood. And so this is work that's been really led by Dr. Nancy Parmalee and Dr. Carrie Heike. And so, we still, again, this is just sort of a work in progress. Okay, and so then I will end with just the future directions for looking at mosaicism more broadly. I think I've laid the groundwork for why I think that really cataloging and understanding the rates and distribution of somatic mutations across many different normal human tissues will give us a search space, I think, for understanding many other human diseases. And pretty recently, we have started working on a project, which is a collaboration between myself, Dr. Andrew Sturgachis, and Evan Eichler, and we have a scientific program manager, Jeffrey Wu, that is called Mosaicism in Human Tissues from Telomere to Telomere. And this is a part of a large NIH-sponsored consortium called the Somatic Mosaicism Across Human Tissues, or SMaHT network. I think it's kind of an obnoxious acronym, but definitely comes from Boston. And so what we propose to do is pretty simple and straightforward. Again, not rocket science, but we're just going to perform very deep sequencing of 10 to 12 different tissues from a total of 150 donors at autopsy over a 5-year period. We're not just doing this here ourselves. There are many other centers across the United States who are participating in this. Here are some of the tissues that have so far been selected. What we locally have proposed to do is about 2,000x coverage using Illumina short-read sequencing in total, and 300x long-read sequencing. You can see 200x Illumina short-read sequencing per tissue, as well as 30x PacBio and 30x Oxford Nanopore sequencing. We are aiming to collect individuals of various different ages. Hopefully about half will be less than 65 years of age, half over 65 years of age. It's much easier to recruit organs from older individuals than from younger individuals. This is not focusing on any specific disease, although certainly many of these individuals will have diseases, and we will record that and make note of that. The only real exclusion is if they have active cancer at the time of their death. we don't want that, because the goal of this is to identify somatic mutation in non-neoplastic tissues. So, and we proposed, obviously with the influence of Evan Eichler, to do complete telomere to telomere genomes under the thinking that the unmappable regions of the genome have some of the highest rates of somatic mutation. And so this is just a picture showing these large repeat elements, which were identified recently as a part of this telomere to telomere project. You can see this enrichment in segmental duplications and a lot of centromeres. But even though these regions are often ignored in a lot of clinical diagnostic sequencing, they do have disease implications, and I do believe, for the exact same reasons they're hard for us to sequence, they're hard for the cell to accurately duplicate, and so they are probably some of the regions that will have the highest rates of somatic mutation. So I am going to end there. I have a few minutes left, and I just want to say thank you again to everyone, all the members of the Seattle Children's Molecular Lab, all the members of the Bennett Lab, and I just want to give a special thanks once again to Dr. Jonathan Perkins, who heads our Vascular Anomalies group, and none of this would have been possible without him, and also just a terrific person and a great mentor of mine, Dr. Mike Astian. I thank you very much, sincerely. It's just been great to work with you and know you. And then, and lastly, I implore everyone to please go ahead and do your teaching evaluations. There's a UPC code for you. I'll be happy to take questions. Thank you very much.
Well, I'll start with some online questions.
Okay.
Colin Pritchard says, amazing work, Jimmy. For the cfDNA approach, have you tried working with GRAIL or similar using a multi-cancer early detection test? Might be even more sensitive, and they might be interested in trying their assays for this purpose.
That's a great suggestion. Thanks, Colin. Yeah, I think GRAIL looks not just at sequence, but also at methylation of these molecules as well, incorporates some I think that's what they do. I haven't spoken with them directly, but that's a great suggestion. I will say, Colin, long before we started doing the cell-free DNA, I went to Dr. Pritchard and said, he sort of helped me down this road a little bit, and so that's a good suggestion. I think you please.
So putting on my skepticism hat just for a second, because I really liked the talk. Skepticism hat in place. What, if any, data is there supporting that targeted therapies to the lesion that you identify is superior to empiric therapy with just an anti-mTOR, with an mTOR inhibitor?
Right. Okay. So I was told to repeat the question. Thank you. That's a good question. The question is, what evidence is that these targeted therapies, such as a PIK3CA inhibitor, alpelisib, or MEK inhibitor, are superior to an empiric therapy, when you're specifically referring to an empiric therapy as sirolimus, let's say?
Sirolimus, most distal part of that.
Right. It's still early days, so I think probably right now there's not a lot of evidence, is the simple answer to your question. And we don't like to put, I don't know that I would necessarily classify sirolimus as standard of care therapy, really. I think actually sirolimus is also targeted therapy. I think it's an important question, is alpelisib, or PIK3CA superior to sirolimus? Obviously, we have a lot more data on sirolimus. It's been treated in children for immune rejection, to prevent as an immunosuppressant for years. And it's well tolerated. But yeah, I think more study is needed. We, as a group at Seattle Children's, we don't like to put any kid on a drug, whether it's sirolimus or anything, without some good molecular diagnosis, particularly because when you put these kids on this drug, there's no good solid endpoint for when you're going to take them off. So they could be on it for quite a long time. Anecdotally, there are certainly patients who've had PIK3CA overgrowth who have responded better on alpelisib than sirolimus. I'm aware of those patients, but yes, there's not yet been like a head-to-head trial. I think another question that is important to ask is that if we're going to say that I'll add to your question, also putting on my skepticism hat, if 80 to 90% of these lymphatic malformations have PIK3CA mutations, then some fraction of the undetected ones will also have PIK3CA mutations. So why not just put them all on the drug? That's something that could be considered. I think if we had really good evidence that the drug was amazing, we might get to that point, but we're not. I don't want to do that at this point. I'm actually a little bit. I have a problem with Novartis because the requirements for going on this drug in Novartis have not included a PIK3CA mutation. And my problem with that is that in these early days of collecting data, if you have a heterogeneous group and you get a weird outcome, then you won't be able to interpret it. So I think at least right now, it's better to be more conservative and test them all, and then as we go forward, we might not need to keep doing that.
Jimmy, really wonderful work. Thanks so much. So I love the first part of your talk, thinking about just all of the different mosaicism that's out there. You had your vascular malformations in a different example. What other examples should we be thinking of in terms of mosaic mutation? I know you've thought about this more than I have, and what other types should we be thinking? Could that be something mosaic?
So there are some classic rules for the sort of the lowest hanging fruit mosaicism. I mean, there are disorders that are known as sort of obligatorily mosaic. So disorders where you see great asymmetry, lateralized overgrowth, disorders that have a predisposition to cancer, disorders that are never seen running in families. This is consistent with something called a haplohypothesis, right? So there's this idea that this PIK3CA mutations that I'm talking about would be embryonic lethal if it was present in a sperm or an egg. And so the only way these sort of super high activating mutations can survive in humans is by postzygotic mosaicism. So I think there are a handful of disorders, and classic disorders, Proteus syndrome is some of these kind of ones. But I think that's the low-hanging fruit for mosaicism. Your question, Brian, is a good one, which is like, what other disorders should we go for? And I don't think we really have a good. We chose craniofacial microsomia. I don't know if it was a good choice. It's an undergrowth of the jaw, right? So maybe that was a bad choice, because you'd think an undergrowth, maybe recurrent activating mutations are more likely to be seen in things that are growth promoting. I think from a pure, just strategic point of view, a disorder that's relatively common, that you see does not run in families, and for which tissue is available. Those are good things to at least start thinking about it. Certainly there are plenty of examples of individuals with neurofibromatosis type 1 that are mosaic. There are a lot of examples of sort of kind of trivial examples of mosaicism that can also run autosomal dominant. But I think the most exciting thing would be like, some of these common isolated structural birth defects, talipes equinovarus, congenital heart defects, and then to go on further, adult onset neurodegenerative conditions, which a lot. There are certainly constitutional rare forms, early onset Alzheimer's and things like that. But there's a lot of somatic mutation in the brain. Could some of that be contributing? Could undetected somatic brain mutations be contributing to some of that disease as well? I think those would be some things to think about.
And are you looking at those in your autopsy? In your sequence every tissue study?
Yes. Sorry, and I don't think I repeated Brian Schertz's question. He was asking in general about different disorders that might be related to mosaicism, and then the follow-up question is, as a part of the SMaHT network, are we going to look at Alzheimer's or Parkinson's, things like that? We're not going to we're not collecting individuals with Alzheimer's disease or Parkinsonism, but undoubtedly, if we're going to collect 150 individuals, at least half of which are over the age of 65, some will have some neurodegenerative dementias. And so we're collecting their clinical history. And so it will be very interesting to see is the distribution, the burden. There have been some studies in people who have looked at this in mitochondrial DNA, and that people have looked at some of these things. But certainly, I think, we're very interested in that. 150 individuals is not that many. And so I think this is just the first step. And so what we'll learn is sort of the search space, and then perhaps from there, if we find, one of the questions is it worth sequencing 5 different brain tissues from 150 individuals to look for these hidden variations? So that's probably where we're going to start to see if it's worth more study. Yes.
Yeah, I was wondering if you had an explanation or a hypothesis for why most of the mutations you find are repeat mutations and not novel.
Yes. So, I think that's certainly, well, okay. I think that the reason is in this pathway, of course, we have oncogenes, growth promoting, which are typically recurrent activating type mutations. And then there are loss of function mutations. We have detected mosaic loss of function mutations in P10, in RASA1, in the same pathway. It's much harder to detect loss of function mutations because they can be anywhere. So I think it's a little bit, we've stacked the deck a little bit. It's easier to detect, when you look in next generation sequencing data and you see 4 alternate reads out of 1,000, but it's at PIK3CA H1047, your power to detect it is higher. So I don't think, I think that it's easier to detect somatic mutations that have a clonal proliferative advantage which is clearly the case in many of these. You know, if you take these cells out in the dish and you grow wild type next to PIK3CA mutant cells, the PIK3CA mutant cells will win out. So some of it is just sort of multicellular competition of cells winning out. But I mean, it kind of keeps me up. I mean, yeah, I think the point you're bringing up is a good one. Novel loss of function mutations that we've not seen before can certainly be present in the mosaic state, but it's much harder to detect them confidently and know that they're not sequencing artifacts. Yeah.
We have a bunch of online questions, but I think in the interest of time, we should probably stop. So
All right.
thank you very much.
Thank you very much. I'm happy to take any questions, follow up if you like afterwards.